Hello students, welcome to Molecular Biology Part 8. In this part, we will discuss about replication. So previously, we discussed about the replication types, and in that proof, experimental proof for semi-conservative replication. Now, in this part, we will discuss about complete mechanism of the DNA replication. DNA replication in eukaryotes. Introduction. Eukaryotes contain giant DNA molecule, and so each chromosome has hundreds of different origins of replication. As the DNA molecule is very long, the strands of DNA cannot be separated to its entire length due to very high energy requirement. Each individual zone of a chromosome replicates as a separate section called replication unit or replicon. I'm repeating again. Each individual zone of a chromosome replicates as a separate section called a replication unit or replicon. Each replication unit or replicon has its own origin of replication. Multiple replicon units are seen undergoing replication at any given time. The advantage of having multiple origins of replication is the speed of this replication process. Replication takes about 8 hours in a human cell. If there was only one origin, it will take 100 times longer to complete its replication. Let's see that a description what we studied in previous slides in a picture module. So what we said, the DNA is a very long in human cell or maybe eukaryotes. Already we studied that in human cell we studied that DNA is 2.2 meters in length and it is accumulated in the nucleus. Its diameter is 10 to the power of minus 6 meters. This we studied during packaging of DNA in eukaryote, that is nucleosome structure. We studied that. So that means in human cell the DNA length is almost 2.2 meters in length, that we know well. So to complete a replication it requires 8 hours. So then this much of a 2.2 meters length, it takes several hundred times it is mentioned in that point. Let's see what we studied, replication unit origin. So in a DNA there are multiple origins. So here origin 1, three origins it is represented in the picture. Multiple origins we can observe. Why multiple origins are starts in DNA replication in eukaryotes? Because of its length. So multiple origins. So that we will call as the replication unit or maybe replicon. Okay. So now, so for each replicon to complete its replication, it takes 8 hours. And also we discussed that why the multiple replication units or replicons are originated in DNA, because it accelerates its speed. It accelerates its speed. So multiple origins accelerate the speed of replication process. As a result, it completes very quickly. Otherwise, it takes 100 times of these 8 hours. That means 800 hours it takes to complete its DNA. Okay. Now, this replication, where the origin region, it is first unwinded. That means the DNA original strands are mentioned here in brown color, and newly synthesized strands are mentioned in the black color. So now these the first old strands are separated and form as unwinding. It will split the hydrogen bonds and the strands are separated, and that new strands are synthesized. So this end it is extended towards this region, and this end it is extended towards this region. So gradually the entire DNA is synthesized as a new molecule. So several origins are starts, and to continue the replication process, and finally the entire length will be completed. So replication is not takes place at a time to the entire DNA length. It starts at several origins and it is extends, and then the replication will be completed. Requirements for replication. So what is required to complete that replications? Just we said that it takes only 8 hours and several origins are formed, that each one we will call as replication unit or replicon. But what are required to complete that replication formation of new DNA molecule? So the requirement, first one, nucleotide pool is required. Nucleotides are required. So replication of DNA requires 4 types of nucleotides that we know very well. That is deoxyadenylic acid, deoxyadenylic acid, and deoxyguanylic acid, deoxycytidyllic acid, deoxythymidyllic acid. These are all nucleotide names. So where they are present, all these 4 nucleotides are present in nucleoplasm. Nucleoplasm in the form of nucleotide pool. Remember it. So nucleotide, because a newly synthesized DNA requires nucleotides. We are all very well known that nucleotide is equal to how it is formed. Nucleotide is nothing but sugar molecule plus nitrogen base plus phosphorus, together called as nucleotide. So the sugar molecule is deoxyribose. The nitrogen bases may be four types, that all we know, that is adenine, guanine, cytosine, thymine. These are nitrogen bases, and phosphate or phosphoric acid. Okay, all these group together to form a nucleotide. So that nucleotides are four types. All four types of nucleotides are present freely in nucleoplasm. Freely. So that free nucleotides used by DNA to synthesize a new DNA molecule. Next, second one, enzymes. Compulsory. Any process in a living organism is regulated by or catalyzed by enzymes. So replication of DNA requires several enzymes which perform a specific functions. So let's see what are the enzymes involved in replication. So that is first one, topoisomerases. These enzymes are concerned with the breaking and sealing of DNA strand. Just now we saw replication, that means replicons are formed. We saw that DNA molecule is like this, and in some region it is separated. It form like this. So here we will call as a origin region. So this is a origin region, and here it is separated by breaking of hydrogen bond. That breaking and resealing, that break ends are resealed again by these topoisomerases. So topoisomerases are required to break DNA strands. That means to break that hydrogen bonds present between the DNA strands in a DNA molecule. That is first enzyme function. Next, helicases. These enzymes unwind the helix of DNA. So between two strands, hydrogen bonds are broken by topomerases, and then its coiling is unwinded, removed. Coiling is removed by helicases. Next enzyme, that is DNA polymerase 1. It is also known as a Korenberg enzyme because it was discovered by Korenberg scientist in 1955. This enzyme participates in the repair of DNA by catalyzing the addition of mononucleotide units to the free 3-hydroxyl end of DNA chain. So it participate in repairing of DNA. How it will repair? By addition of mononucleotide. We are studied that deoxyribonucleotides monophosphate. Similarly, deoxyriboguanine monophosphate, deoxyribo monophosphate, cytosine monophosphate, deoxyribo thymidine monophosphate. These units are added at 3 end. So 3 end consists OH group, hydroxyl group. So this any one of these, not all four at a time, any one of this mononucleotide is added to this hydroxyl end or third end of the DNA chain. That it is regulated by DNA polymerase 1. Next, another enzyme, that is DNA polymerase 2. So the biochemical role of this enzyme is not yet known. So that's why in our discussion of the mechanism of replication, this enzyme will not be mentioned. So remember why it is, because not yet known, and it cannot replicate the long strands of DNA. Okay, so we in exam or maybe anytime we need not to remember more, but if anyone asks, DNA polymerase 1 is there and 3 also is there, but what about 2? So 2 not at known. DNA polymerase 2, its function is not at known. Next, DNA polymerase 3. This enzyme was discovered by T. Korenberg and M.L. Jefter in 1972. Korenberg discovered DNA polymerase 1 in 1955 and this polymerase 3 in 1972. It is mainly responsible for DNA chain elongation. Chain elongation means, already we said, a DNA molecule is unwinded, break and unwinded by topomerases and helicases. Okay, so now here a new strand, new DNA strand is formed to this, parallelly to this. So it is initiated like this and it is elongated. The newly synthesized strand is elongated parallel to old strand. That elongation of this DNA, new DNA molecule, regulated by DNA polymerase 3, that is main function. Okay, remember DNA chain elongation is regulated by DNA polymerase 3. Next, this DNA polymerase 3 catalyzes the replication of DNA in 5 to 3 direction on 3 to 5 strand. So we are all very well known that the DNA consists two strands, one strand, one strand one strand on 5 to 3 direction, and another strand is anti-parallel to that, anti-parallel, that is it is present on 3 to 5 direction. Clear? 3 to 5. So now what the point says, that the replication of DNA in 5 to 3 direction on 3 to 5 direction, on 3 to 5 direction. So 3 to 5 direction is 3 to 5. So now this is from 3 to 5, it is above. That means the replication, suppose it acts as a, this strand acts as a template. That means old strand, it is separated. Now newly strand is formed. Suppose it is third end, then the new strand is starts to form 5 to 3, because it is a new one, sorry, old one, and a new strand need to form parallel to this. That means here it is the old strand having 3 end, That means a new strand, it starts a fifth end here. Anti-parallel. Then the newly formed DNA is always 5 to 3 direction. 5 to 3 direction. And suppose if you take this one, this is also same. It is not start from here. Again, it starts from here. That means here third end is there. On this, the newly synthesized DNA is initiates a third end and it is extended. So this is a 3, then newly formed and having 5 end. So the DNA replication is always a 5 to 3 direction only. See, 5 to 3 here and here also 5 to 3 direction on 3 to 5 strand. So it never changes. So please remember this point clearly. The DNA replication or newly synthesized strand is always 5 to 3 direction on 3 to 5 strand of DNA. Next enzyme F, RNA primase enzyme. This enzyme is also called as RNA polymerase. It is very essential. It is concerned with a synthesis of short segments of RNA called RNA primers. RNA primers. So this RNA primase is required to synthesize a short segments of DNA. These short segments of DNA called as RNA primer. Once recollect, we studied previously about genetic material. DNA acts as a genetic material because it is a stable in a chemical nature, stable in structure, and also it has a stability due to presence of a thymine instead of uracil, and it stores the information. Is it clear? We have studied this point clearly in previous videos. So DNA has ability to store the information, but it needs RNA to transfer that information. RNA is compulsory. It has ability to store, but RNA has the ability to transfer that information. That's why we required both. So now to replication, what it requires to bring nucleotides to join to form a new chain. To bring that nucleotides, RNA is required. So that's why it RNA primase first forms that RNA primers. Until the formation of RNA primers, the replication does not initiate. Next enzyme, DNA ligase. This enzyme uses ATP. This enzyme uses ATP energy to join the small fragments by sealing the sugar phosphate backbone. Sealing the sugar phosphate backbone. So ligase is required to join the small fragments. The next third requirement. Till now we discussed enzymes. Now third one is RNA primer. Already this RNA primer is synthesized by RNA primase or RNA polymerase enzyme. Now what is RNA primer? It is a short RNA segment that is found on DNA template before the replication can begin. Before replication, this RNA segment, a small segment of RNA is formed. That segment or that RNA primer initiate the process of replication. So RNA primer is essential to initiate the process of DNA replication. So see here the requirements what we discuss in that diagrammatic representation. See here, just we are taken the part of unwinding area. So here topoisomerases. So it breaks the hydrogen. These are all, these lines are the hydrogen bonds are broken. And then this helicase, this green circle, helicase is it unwinds that region, unwinds the two strands. So now each strand, this is a one DNA strand, that is a parent DNA strand, we will consider as template. Template, it is a old DNA strand, based on this new DNA molecule is synthesized, and it is also acts as a one more template. Template, it is a old DNA strand, old DNA strand. Based on that, the new DNA molecule is synthesized. Two strands. So on this, see this one template having this 3 to 5 direction. That's why newly synthesized DNA, newly synthesized this one, always 5 to 3 direction. And similarly, one more DNA molecule is formed here. It is also 5 to 3 direction. So formation of new DNA is always 5 to 3 direction on 3 to 5 strand. Now, what is required? RNA primer, primase, RNA primer. First, on this old strand, a small segment of RNA primer is required. RNA primer is formed. So here a sequence is present, nucleotide sequence are present here. It is already, you know, that nitrogen basis nucleotide sequence. So this old strand having a nucleotide sequence. That means a nucleotide with A here. That means it consists A, it consists C, C, T, G, like this. That means the nitrogen base in this nucleotide here, cytosine, and here it is A, adenine. That means this side the new strand should have, it is A, no, adenine, no. The new nucleotide should have thymine. That where it will get the nucleotide with this thymine, nitrogen base, in the nucleoplasm. Already we studied that. First requirement is nucleotides only. The nucleotides are present in nucleoplasm, in nucleus itself. So now this RNA primer, according to the sequence on the old strand, it collects that nucleotides from the nucleotide pool and brings here. Then strand is extended. So primer function is as per the sequence present on this template strand, it joins that nucleotides. That's why see 5 to 3 end, it is 5. That means here it is 3 end. No? 3 end is having hydroxyl group. That's why it is reactive. The new nucleotide is added. It is synthesized. 5 to 3 direction. Why it is always occurred in 5 to 3 direction? 5 and always having phosphate is not reactive. That's why the nucleotide is added to the hydroxyl group that is present always on third end. That's why it is always carries 5 to 3 direction, new molecule synthesis. It is also one important point. Why it is occurs only 5 to 3 direction only? Because third end consists hydroxyl group. It is reactive. So that's why newly formed nucleotide is added to this third end. Okay. So now DNA polymerase 3, it continues that reaction. That means it elongates. The nucleotide elongation will continue, elongates the chain. Okay. Now, mechanism of replication. The initiation of replication, that is uncoiling or maybe unwinding of the DNA double helix, begins from a particular point which is called as origin of replication or maybe replication unit or replicon. There is only one origin of replication in E. coli, that means in bacteria, prokaryotes. In prokaryotes, only one origin. Okay. There are several origin of replication in eukaryotes, multiple origins. Already we discussed in the starting itself. Now replication may be unidirectional or bidirectional. So see here, it is a unwinding, uncoiling of this DNA strand, uncoiling the expanded region. So this region, it is a normal DNA, and here. So now the replication is bidirectional or unidirectional. It is mentioned. So the DNA moves this and also it is moves. So 5 to 3 direction. See, it is a 3 to 5 strand. It mentioned 5 to 3 here. It cut. This part is cut. That's why it is 5 to. This is a mentioned. It will come coiled. No, it will come this one. Actually, it is like this, and this is like this. That's why it is 5 to 3. Be careful. So now see here, small gaps are there. These hydrogen bonds are broken. These hydrogen bonds are broken by helicages, and here it is sealed, and it is uncoiling. Is sealing is done by helicase, and it is uncoiled or winding. This broken end is winding. This total winding is done by helicages. Helicages are winding this one. So now in this region, it occurs in bidirectional replication that already we discussed, unwinding. So where this replication is starts, that region. So this region we will consider as the origin of replication or replication unit or replicon. The process of DNA replication involves the following steps. Step 1, the activation of deoxyribonucleotides. First, nucleotides need to be activated. So the nucleotides occurs freely in nucleoplasm. Already we mentioned this point. They serves as a raw material in DNA synthesis. So the raw material for this new DNA synthesis is nucleotides. They are present freely in nucleoplasm. Nucleoplasm is nothing but a fluid substance present in the nucleus like cytoplasm. Nucleotides are activated by phosphorylation, addition of phosphate with the utilization of ATP. ATP having triphosphates. So nucleotides are activated by phosphorylation. Once check that we are given the types of nucleotides table in the early videos. There we are mentioned deoxyribo, adenine, monophosphate, deoxyribo, guanine monophosphate, deoxyribo, cytidine or cytosine monophosphate, deoxyribo, thymidine monophosphate. These four types we are mentioned in a tabular form. Once recollect. So these are all in monophosphate, having one phosphate, monophosphate. When they are utilized this ATP and it is phosphorylated, means phosphate. From ATP, phosphate is added to these molecules. Then they become triphosphates. Deoxyribo, adenine triphosphate, deoxyribo, guanine triphosphate, right? It become ATP, GTP, nucleotides are become CTP and TTP due to phosphorylation. And this phosphorylation, addition of phosphate to these monophosphates of nucleotides catalyzed by phosphorylase enzyme. Second step, unwinding or uncoiling of DNA double helix. Before replication starts, the enzyme helicases and topoisomerases take part in unwinding of DNA helix, both together. Helicase function is recognize the specific origin of replication and cause unwinding of the DNA helix. And this topoisomerase, this is also called DNA gyrase, break and reseal one strand of DNA to facilitate uncoiling. So what it breaks? It breaks the hydrogen bonds between the strands. So let's see. So it is a first one. It is a total DNA. In that several, that means many origin of replications are formed. So it is a origin. So this origin of replication is identified by the helicase enzyme, and these hydrogen bonds are broken by topoisomerase or maybe DNA gyrase enzyme. Now the replication is proceeds. The separated strands are stabilized by they are stable, not rewinding again. They are stable in that uncoiling position by helix destabilizing proteins. They stabilize this unwinding double helix strands. These proteins keep the two strands separated for replication. The separation of the two strands proceeds slowly from one end to another. It results in the formation of Y-shaped structure called replication fork. The two separated DNA strands function a templates, that means a old DNA on which the new DNA strands are formed. Third step, formation of RNA primer. So see, till now, first step, phosphorylation of nucleotides. Second step, unwinding of double helix. And then next step is formation of RNA primer. It is essential to initiate the DNA synthesis. So already we mentioned RNA primer is a short segment of RNA with 5 phosphate at 5 end and hydroxyl group at third end. Unwinding of DNA is followed by synthesis of RNA primer of 8 to 10 nucleotides. Remember, how many nucleotides are present in a single RNA primer? That is 8 to 10 nucleotides. On template strand, this RNA primer formed based on the sequence of that template strand at the end, that is at the 5 end region. The RNA primer is synthesized with the help of an enzyme RNA primase or polymerase enzyme. The formation of RNA primer constitute the initiation phase of the DNA replication. RNA primer is must for the synthesis of a new strand of DNA. So once see the two strands are separated, it is appears like a Y shape. So see, this is if strands draw like this, it is unwinded strand 1 and another strand. Then it is appears like a Y letter. So this one is called as replication fork. So it is a replication fork. So in this, the initiation is copied by initiation is starts by C here. Fifth end, it is started with the formation of a primer. Until unless if there is no primer, there is no initiation of DNA molecule. So now, so topoisomerase unwinding by breaking these hydrogen bonds. That means break the hydrogen bonds, and then this helicase unwind, separates, uncoil these two strands, and it form as a Y shape called as replication fork. And then the next step is starting of the clamp means it is seal. So starting the here it initiates on the 5 end, it initiates first a primer, RNA primer, and that RNA primer, what it will do? RNA primer initiates the strand, and also we are stabilizing protein, we said. So that is a single strand binding protein. So these are called as the destabilizing proteins. These proteins keep this uncoiled. These proteins keep these unwinding strands in their position. They are not allowed to recoil again till completion of replication. Base pairing. Already we discussed about this base pairing according to Chagov rule. So purines always pair with the pyrimidines, or simply we can say adenine always pairs with thymine, guanine always pairs with cytosine. This we know. So now the RNA primer formed is removed and the gap is filled with deoxyribonucleotide. So RNA primer formed first 8 to 10 nucleotides formed with that RNA primer. That primer is removed and that gap is filled with deoxyribonucleotide to initiate the formation of new strand of DNA, and it is catalyzed by DNA polymerase 1. The replacement of RNA primer by DNA nucleotide is proceeded by the pairing of appropriate deoxyribonucleotides, that is ATP opposite to T. A adenine triphosphate, adenine it is a nucleotide opposite to thymine triphosphate, thymine. Guanine triphosphate opposite to guanine bound with cytosine, and thymine opposite to adenine, cytosine opposite to guanine. Like this, the appropriate pairing is formed, and this is initiated, catalyzed by. So, replacement of that RNA primer with the DNA nucleotides, deoxyribonucleotides. RNA primer is formed by ribose nucleotides. That ribose nucleotides are replaced by deoxyribonucleotides, that is initiated and catalyzed by DNA polymerase 1. The two extra phosphate present on the deoxyribonucleotides are set free with the help of an enzyme pyrophosphatase, and the energy is released, and this energy is used for the formation of hydrogen bonds between complementary bases. Remember. So now unwinding one strand, take only one strand. Actually two strands are there, but we can take one strand unwinding, and on that first it forms a primer. Clear? A primer is formed, and this primer is replaced by deoxyribonucleotides. It is a primer is a RNA. RNA having ribose nucleotides. You know very well, because the sugar is ribose sugar, not deoxyribose. Ribose sugar in RNA. These ribonucleotides are. So now what happens? This RNA having a sequence. Am I correct? RNA having sequence. No, RNA also it is a nucleotide chain. That means suppose the 8 to 10 nucleotides, we are saying that this RNA primer is a 8 to 10 nucleotide sequence. 8 to 10 nucleotide. So one nucleotide A, another nucleotide U, another nucleotide G, or another like that. You can take a G, like this nucleotides are present, and it is, remember, it is a RNA primer. It is a primer RNA. Now what happens? It need to replace by deoxyribonucleotides with the appropriate base pairing. Correct. This A bonds with T. Okay. And U, that means A. Uracil bonds with GC. GC, like this. And it joins here. It is entirely replaced with deoxyribonucleotides. And then it is initiates with a synthesis of a new molecule like this. Then it is continue with deoxyribonucleotides. Okay, now what we said, the nucleotides, first it is phosphorylated. That means it become triphosphates, ATP, GTP, CTP, and TTP it forms. So that means when these triphosphates are joined in this nucleotide formation, it loses two phosphates. Triphosphates, no? Only DAMP is added here. Monophosphate. What about remaining two? They become free. So that is free with the help of an enzyme pyrophosphatase. That means when the phosphates are released, become free, that is nothing but it releases the energy. Remember that one. Energy, free energy is produced. Now, this free energy, when the two phosphates are separated, whatever the energy is released, that energy is used for the formation of hydrogen bonds between between two DNA strands. So see this, the replication. So now here it is unwinding, and this is the region of the origin of replication. This is the point of origin. So from this point, one DNA molecule, that means new DNA are synthesized in this direction. Another one is synthesized in another direction, bidirectional. So now see, initiated by here this color. This is a primer. This RNA primer. And then the chain is extended 5 to 3 direction. So by addition of that nucleotides, what we said here, when this nucleotide, this is all nucleoplasm, it is totally, the surrounding is a cytoplasm present in the nucleus called as nucleoplasm. The fluid substance present in the nucleus is called nucleoplasm. So in this nucleoplasm, previously deoxyriboadenine monophosphate, initially it converts by addition of phosphates, and it become ATP. Now these are used to form new DNA. So when this is added here in a DNA synthesis, this triphosphate loses two phosphates and only DAMP is added. And this released phosphate, when the two phosphates are separated, we studied that when ATP is splits, it releases the phosphates plus energy, because bonds are broken. When bonds are broken, energy is released. So that energy is used to form these hydrogen bonds between the two strands. Initiation and elongation of DNA strand. It is the last step. Initiation and elongation of DNA strand. Formation of new strands of DNA is made 5 to 3 direction from 3 to 5 template in the presence of an enzyme DNA polymerase 3 and magnesium ions are required. The polymerization of nucleotide, that means addition of more number of nucleotides, takes place only 5 to 3 direction. The replication process occurs on one strand in one direction, and on another strand it occurs in opposite direction. That's why it is bidirectional. Next, in one strand, the synthesis of new strand, the synthesis of new strand goes on continuously in 5 to 3 direction, and this new strand is called as a leading strand, and such type of replication is called as continuous replication, is occurred on one strand. On the other hand, short DNA segments are formed. Short means small segments or fragments are formed in 5 to 3 direction. On other hand, two strands are separated. No? On one strand, DNA is formed continuously, and another strand. So let's see here. Suppose this is a one strand. It is 3 to 5. One strand. On this one strand, the DNA form 5 to 3 direction. DNA form continuously. Continuously. And another strand, on another strand, this is another template, parental DNA. So it is 3 to 5. Another strand, the form, DNA forms in a small fragments. So the continue form, this one we will call continuous replication. And another strand, it forms very a small or maybe a short DNA fragments or segments are formed in 5 to 3 direction. 3 direction. Why it is formed like that? Why it is not continuous? Due to exposure of a small stretch of another template at one time. Due to exposure of small stretch of another template at one time. So one time the exposure is only small segment. That's why it forms only small parts. And these short segments are called as Okazaki fragments or maybe Okazaki segments. Now, already we discussed RNA primer is essential for the synthesis of new DNA. Yes. Now, on this second part, that means where the fragments are formed, small fragment, each DNA fragment required one RNA primer. So, an RNA primer is required every time a new Okazaki fragment RNA primer. That means here one primer is from RNA primer and here RNA primer, and again if any fragment, then again it is initiated with RNA primer. Whereas in continuous strand, once RNA primer is formed, that will continue the initiation and elongation of DNA chain. But here every fragment is initiated with a RNA primer. The RNA, after formation of all, after completion of one replication here and another strand, fragments are formed. So the RNA primers are then removed and the gaps are filled by DNA synthesis, by DNA polymerase 1. So these primers are removed and DNA polymerase 1 is involved to join that nucleotides in this. And nucleotides are joined by DNA polymerase, and sugar and phosphate bone is joined by DNA ligase. Gaps filled by DNA polymerase filling, and it is cementing. Joining is by DNA ligase. So this strands is called, the Wakajaki segments formation strand is called as lagging strand, and the mechanism is referred as semi-discontinuous replication. So observe this photograph clearly. The entire replication process you can understand now clearly. And the points are also mentioned. Helicases unwind the parental double helix. Single stranded binding proteins stabilize the unwinding parental DNA. And the leading strand is synthesized continuously, that is a leading strand by DNA polymerase enzyme. And lagging strand is synthesized discontinuously. The primase synthesizes a short RNA primers for everyone, and these segments are called as Wakajaki segments. And later the Wakajaki RNA primer is replaced by DNA polymerase 1, and ligase joins the Wakajaki fragments. That joining is by, so the whatever we discuss briefly we are mentioned in this picture. The one more picture, the representation of this DNA replication. Sir, what is this one? Already we shown now. Yes, this is also we shown a different turnings and all, because you can remember that the DNA is highly coiled now as a nucleosome. So if you are see, it is appear like this. So simplifying diagrams are very easy to see. Actually, it is very difficult. So now SSB, wherever you can observe that SSB, single stranded binding proteins, they hold the unwinding strands in their position. Okay. So primase, that means an enzyme. This primase is helpful to synthesize RNA primer. Okay. So DNA polymerase elongation of that DNA molecule. Actually, this is here one part, here one part, and here. That means it is a lagging strand. Okazaki segments. These are all, and this is the leading strand. Continuous replication. This one is the discontinuous replication. Continuous replication in this direction, and discontinuous replication is, that's why it is bidirectional. Okay. So now you may have doubt, beta clamp alpha, that means beta is there, sigma is there, sigma is there, and these are all the parts of DNA polymerase 3, remember. Okay, so now this is a brief representation. Previous slide, it is a very simple diagram to understand, and it is somewhat a difficult part. Okay, so but if you listen again and again, at least listen one twice. Okay, so after listening a few minutes, once go through the textbook, then you can understand clearly. Okay, so it is a very important essay in PU board examination.